welcome to Microbiology Twitter Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life I dropped out for my PhD in microbiology at the University of Chicago, where I grew with MRSA. Nowadays I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to enabling the public with the tools of biotech.
My name is Faz, and I have a PhD in microbiology, and I've mostly worked on bacteria, but I also worked in research integrity afterwards, and now I'm currently working as an academic editor for a scientific journal.
So today is our news week. Each week we meet to talk about microbiology, and in this week we do an overview of different papers that we've seen in the news or other sources to try to find an article that we want to cover next week in more detail.
Yeah, and next week will be our deep dive week, where we take the article we choose today and look at it figure by figure, detail by detail, to learn what it can and cannot tell us. So make sure to subscribe to satisfy your microbiology curiosities.
And you can follow along with all the papers that we discuss in either week in our shared Zotero library linked in the doobly-doo below.
Yeah, and we want to hear from you, so please use the comments to tweet us, comments or tweet us using the microTWGC hashtag. Yeah, and so let's go on. So this week we'll be looking at vaccine news, because there's been a few bits of news we can pick out from this week. Going to start off with vaccine based news. Yeah, so
Immunity provides more robust heterotypic immunity than natural infection to emerging SARS-CoV-2 variants of concern.
Yes, indeed. So let's see if I can hold up a second. I just need to pull this up on my screen share.
So this is, this is what is on everyone's mind, I feel, right now. There's so many, we'll cover a lot of this week, I think, right? Of how do the vaccines stack up against these variants? Because that is what the media is, maybe not explicitly talking about both together, but yeah, definitely obsessed with both of these elements.
Yeah, so because there is that question of there's been a few suggestions in papers previously that vaccines provide a better immunity than just sort of natural infection. That's because there's papers come out that said that it produces a higher titer of antibodies, and yeah, and
we've seen papers, right, that say that SARS-CoV-2 has proteins and things that inhibit immune responses, right? So if you're getting your immunity through sort of just the purified components, right, or even the dead virus for that matter, that's going to be different than the immunity that you get when the virus is actively trying to antagonize your immune system so that it doesn't get caught.
Yeah, I just remember that paper we did a while ago on B cells, where it impairs, causes some craziness of the B cell memory and stops germinal centers from forming, so the actual way the immune response is formed is slightly impaired in some patients for SARS-CoV-2.
Yep. And then we've seen papers where non-structural proteins inhibit translation, potentially suppressing interferon responses. We've seen that there are changes to, I think we may have done news last week. There was something that said that even the CD8 profiles are a little bit different. So yeah. Stuff is happening, right? This virus isn't just sitting idly waiting for antibodies to be made against it. So this is following up on that intuition, I guess, right? Yeah. That sort of first principles thought that the vaccine will elicit a different immunity, probably a better immunity.
And the idea is that, they test it in people who are infected with the B117 strains and the B1351, so that would be the UK and South Africa strains. And the thing that, we've seen a lot, but what we haven't seen is figure 2F. So I'm going to pull it up on the screen. It'll take you a while to see it, but so they represent the spike protein as a squirrel. So
oh yeah,
yeah. So this is something I've seen is done in a couple of papers recently, but yeah, here they
just a squirrel or just anything.
Not specifically a squirrel, but just anything, something that is similar to that shape, so they can kind of give an idea of where are these antibodies binding to. So so in this case,
they head is maybe more easy to think about than receptor binding domain.
Yeah. So is that mnemonic? So yeah, I was scrolling through this paper and I saw a picture of a squirrel. I'm like, wait a minute, this is a microbiology paper. Why is there a picture of a squirrel? But there it is.
Yeah, it's important, right? A lot of the things that I think people want to know is that the antibodies that you elicit from immunized serum, right? Where are they binding, right? On the on the spike protein, and are those regions, right, the regions that are changing with the variants?
Yeah.
That's the big question right now.
So yeah, this is how they've chosen to show that in a way that is easy to remember. Like they
Yeah,
go for the head or for the tail, I guess, of the squirrel.
yeah, tail, but I'm not sure.
Yeah, I
So like a neck, it seems like they've circled.
yeah. But anyway, so moving on to next paper, which is the safety and efficacy of Charadox nCoV-19 brackets AZD1222
Mhm.
COVID-19 vaccine against the B1351 variant in South Africa. So this is basically another paper from the Charadox group that. You may be aware that those one of the studies was based in South Africa. So this is a report that on that part of the trial, specifically in South Africa, that goes into a bit more detail and tests serum for people and vaccinated people against the B1351 strain, and
Mhm.
yeah, and looking at the kind of purport relating the efficacy of that they found in this study to the 131. So seeing like, is there any difference between this and in other places? So in because you'd expect that if there was some kind of neutralization difference, then this might see some kind of difference in efficacy. Um,
Yeah, we had seen some of those preliminary reports, right, showing that there was a difference in antibody, the neutralizing antibodies that were made, um, in terms of, yeah, if it was able to neutralize the variant strain.
Yeah, so this focuses on it, and I think again it shows that there is a vaccine response, but B1351 is just slightly more resistant. And
Hmm.
if we want to go into more detail about that, this is the paper to read about in relation to
Yeah,
Childux 1.
yeah,
Um,
absolutely. Is this also, I feel like this is part of supporting the news story where they aren't giving the AstraZeneca vaccine out in South Africa, or distribution was limited in some way?
I mean, I didn't hear that news story, but that would make sense if
I
they
see.
limited it because of that. Um,
Mhm.
yeah, I don't know what to. So yeah, I haven't been keeping up too much on like the vaccine kind of
Yeah.
shipping, but yeah, that's. I mean, this strain is there are a lot of questions out there, and I think that we're focusing on the antibodies, and but yeah, there are questions still about
Yeah,
what the
there's
actual
that
effects
other
are.
element of T cell immunity. It might not be the whole story. Hmm.
Yeah, uh,
But yeah, but again, they are seeing in this, they're looking at vaccinated serum versus non-vaccinated serum in this paper, and they do find that there's less neutralizing ability
mm,
for serum.
yeah. So next one, oh, this is an interesting one. Epidemiological and evolutionary considerations of SARS-CoV-2 vaccine dosing regimes. So, you might have
Yeah.
heard like that lots of people are doing single dosing. Lots of countries are starting that to get as many people with the first dose as possible. But there is one
Mhm.
criticism that keeps coming up that if you give people a single dose, it's not going to be perfect immunity. And imperfect immunity can give viruses the opportunity to mutate and to
Yeah.
evolve to resist those vaccines.
But
And
we'll also say even when you get the two doses, it's not perfect immunity, right? Like it's not going to stop every single thing, but I guess like the relative difference, right, is potentially a problem, right? Like it's less.
yes,
Getting one is not as robust of a response, I guess what we could say about the immunity.
yeah, and this paper is a modeling paper, so it basically tries to model game out each of those outcomes. So it goes, what if say the first dose is really effective? What effect does that happen? Or what if the first dose is really ineffective? And then
Yep.
they do like some susceptible, infected, resistant modeling, and they
Yeah.
just build up on
We
that.
covered a similar type of model, right, in construction in a previous episode where we were looking at the emergence of the variant of concern in the UK. So they basically adapt, like it's a theoretically it's a very similar model. Of course,
Mm.
like they have to stratify it into even more versions because they want to do this one dose versus two dose strategy.
Yeah, and once it does that, it does the next step of actually trying to model the evolutionary pressure to become resistant. So
Hmm.
this is like almost two different schools of modeling because evolutionary model and transition modeling don't often meet, but then they put them together in an interesting way, and
Yeah.
and so
I like it. I mean, it's
yeah,
interesting because I feel like we haven't talked about the evolutionary modeling at all,
yeah,
which is what. interested me about this paper because it's like, oh, like there are also statistical dynamics we could say, right, for a virus going along. How does that intersect with our knowledge of who's getting vaccinated? What are the sizes of the infection peaks and stuff like this?
Yeah, exactly. And this games out like different ways of. So it says, okay, if the first dose is very strong, or if it's weak, and they say that okay, if it's very weak, then actually like there might be a chance, but if it's very strong, then the single dosing strategy actually might be very effective, and we don't want to miss out on that. So I think that's an interesting one. I mean, it is still a modeling paper, so there are pinches of salt out for predictability, but in terms of like thinking out like making
Yeah,
predictions.
it's a
Yeah,
conceptual framework for us to
yeah,
understand, right? Like, what are the knobs that are tunable in the system, right? And like, give us a sense of if we did tune this knob, what might we expect to see? Like, I mean,
yeah. I think that so. This is a really fascinating paper. So I think to
mhm.
read and look at. The next one is all right. mRNA 1273 efficacy in a severe COVID-19 model. Attenuated activation of pulmonary immune cells after challenge. So
So this is like I mean, we've said, I hope we've said this before, but like clinical trials show us like a relationship between the use of the vaccine and a clinical endpoint, right? And that clinical endpoint is something relevant, like less disease or something like this. But here we're actually looking at maybe like more of a mechanistic evaluation of the vaccine. Like, what exactly is the vaccine doing that makes the disease less severe?
yeah, so they go into hamsters, because human studies can't provide the controlled response to infection and complex immunological insight that are possible with some preclinical studies. So the test of this study is whether it can provide us with more insight than human studies. So they do things like measure the virus replication in lungs, and then they do this really like I think we've talked about this before, integrated cell analysis, where they take a bunch of like genetic data from hamsters and they put it into a graph and they show you like what difference does vaccination have on these different cell populations and how they are
Yeah,
distributed.
this is like a single cell technique of some sort,
Yeah,
right? Like this is by cell type.
yeah. They
Hmm.
RNA sequenced every cell in that cell type and
Yeah,
then classified and picked out the differences. So you can actually kind of really pick out individual tiny differences based on
right.
whether they were vaccinated or not.
In, I mean, just as a little sidebar to this, like back in the day, right? Like you just be doing a lot of histology to get at this, and
Oh
like
god.
staining all the different slides to find all the different cell types. And here you can take that whole organ as long as you have a technique to dissociate all the cells into single cells, and you have a great panel of markers. You basically get that information without having all of yeah. It's a much more comprehensive view, I think, of infection than we've ever had before.
Yeah, so there's a lot of like potential in this paper, um, and yeah, that seems like it. So I think like moving on to like preclinical models, we're going to be looking at next paper is revealing tissue-specific SARS-CoV-2 infection and host responses using human stem cell derived lung and cerebral organoids.
Yeah. We've had like organoid papers on the docket, I think,
Yeah,
from our choices, but this nice to see this is a published paper, a pre-proof at least, accepted into Cell. And this is one of those things where, it's another space, another model space, right? Where you can models are only as good as like what they exactly correlate to, right, in the real system.
mm.
And one of the ways that you can try to get a correlation between the real system and the model system is to use the same cells, basically. So like these are human cells, but instead of being differentiated organs in a body, they get differentiated in a petri dish into like a more complex sort of tissue-esque object they call an organoid.
Yeah, this is interesting because it's not just like a flat. So usually with cell studies, you imagine like a flat plate of cells that were used to be a part of the lung. These will actually maintain some of the structure of the lung and some of the tissues. So you can do things like, in this case, added the SARS-CoV-2, and then they could
Yep.
like measure like what kind of outputs there were. So uh, basic immune responses from the innate immunity, like what kind of cytokines do lungs have? What cytokines do they produce when they're angry by the Sorry, I'm so tired. I cannot speak properly.
Yeah, no problem. I mean, I'll just say from my experience, right? Like I was working on skin organoids, and so like you can even it's like I think it's very clear to people when you think about the skin. The skin is a lot different than just cells. It's like our skin is not just a layer of cells that lives upon us. But I mean, the same is true for lungs and brains, right? They have some organization to them, and that organization means they express different things, and that means they interact differently with pathogens. So yeah, this seems very exciting. I mean, I just love hearing about where organoids are going these days because I think they're a very important technology, and they can remove our reliance on using so many animals.
Yes. Um, Speaking of that, we've got this paper from Nature called SARS-CoV-2 infection is effectively treated and prevented by EIDD2801. Now,
Yes,
this.
with quite a frightening animal model, just
Oh
like
yeah, this.
from sci-fi, maybe. I'm not sure if people don't read a lot of science papers, maybe you haven't seen some of the types of animal experimental designs that exist out there. Um, yeah, this one's a strange one where they subcutaneously implant human lung tissue into mice.
So yeah, they implant like human lung. Yeah, this is a paper that I am not. So there are like there's this type of animal model where you do this, where you implant like a tissue from a different like organ and then
Mhm,
get some idea about it. Uh,
mhm.
Personally, I'm not a fan of this system because at this point you're using the mouse not as a mouse model but as a petri dish to support your cell culture that you're putting in a mouse.
Yeah.
Um,
Sure. I
but
feel like, I mean, this would be one of the if we chose this paper, this would be one of those things we're looking for. But like, what about mouse cells that infiltrate this tissue, right? Like, what goes on in that
Well,
circumstance?
here's an interesting thing. They usually use immunocompromised mice, so that means they don't have very good immune
Mm,
cells.
mm,
But
mm.
the actual lung implants do have an immune system. They still have like some kind of so
Cells that sit in there, yeah.
but yeah they use this to test out bat coronaviruses and to see whether they replicate in the cells.
Mm.
Again, no adaptive immunity. There's not really. I'm not sure what represents, but they kind of create this SARS-CoV-2 model,
Mhm.
and they measure the kind of pathogenesis that they have in that is very much. So it's very similar to what we saw in the lung organoid paper in terms of like the production of cytokines, the induction of different cell, uh, and
Yeah.
then they test a ribonucleoside analog, EIDE201. So this is basically it seems to be like a model characterization paper, but with like the actual treatment tagged on at the end, just to show we um
Yeah, we've seen this format of paper
hmm,
before. Like when they showed us the human ACE2 mouse,
yeah
it was often tacked on to a monoclonal antibody study at the end, where it was just
yeah
like, and now that we've shown you the model, here's an antibody that works in it to stop the disease. So very similar. Another type of model. I agree with you. Now thinking about it, might not be that great of a model. Only diving in would really tell us the answer to that.
yeah.
Um,
I mean
and yeah, and a potential therapeutic, which they say in the abstract is also in phase 2 and phase 3 trials right now. So
yeah, I mean, it is published in Nature, so there might be something we're missing that is actually really good, but I can't really see it from my brief skimming of it. Um,
okay, let's move on.
yeah, let's move on. Survey of PeriDomestic mammal susceptibility to SARS-CoV-2 infection. So, what do you think of this one?
Well, first of all, I've never heard of the term peridomestic,
Yeah,
but I guess it refers to like city animals.
yep,
Yeah, animals that live in the same habit, like sort of same ecosystem that we live in, the city ecosystem. I assume what it's going for is trying to figure out reservoir stuff, right? This
yeah,
information is going to tell us, is there a significant reservoir in our animals? Should we be? I mean, this is as we progress through this pandemic, right? This was something I remember hearing early, right? Like we could expect that this coronavirus may become a seasonal thing
mm,
if it finds a reservoir somewhere, if we find that it does change at some frequency. And like we may not get severe disease from it anymore or as severe disease as before, but it could exist in some population. So they're looking here at the susceptibility of these animals as potential reservoirs, I
Yeah,
guess.
so like usually with these sorts of surveillance, you'd go around and you'd swab a bunch of animals to see whether they had COVID. But in this case, they actually live caught wild animals and
Mm.
infected them with SARS-CoV-2, which I'm not sure is entirely ethical. I think
Oh,
that
you
that
think
would.
that that's kind of questionable from the IRB perspective?
Yeah, I mean there are like some specific things about like wild caught animals and and the reason being able to justify why you should catch them and why it's necessary to use them in research because usually we use lab caught animals and it's not that so because it's weird because there's this line between like taking stray animals and using them in research that's been banned for a while because it's
Yeah,
um
yeah,
and this is almost straddling the line but I can sort of see the reason for it because it is sort of necessary to understand what animals would be susceptible, what animals will carry this coronavirus if we clear it out of humans, what animals will we
yeah.
potentially have to think about vaccinating or treating to stop coronavirus
Well, I
occurring
think it's
even.
great that you brought up that point because it's almost an entry point into this discussion of like the role of IRBs,
Yeah,
right? And like where, like in what ways do we justify animal studies, right? Because I think that for sure during this pandemic, I'm sure there have been studies that have been justified in animals that like maybe wouldn't have been previously just because of how the scale of the human cost of the pandemic, right? And like people are like, well, this justifies, right, use
well,
of animals in this way, perhaps.
yeah. You always think about like how many animals to save a human, or like how many humans are dying per second versus and you kind of like think of, and with the SARS-CoV-2 outbreak, so many humans are dying of this disease that
Mm-hmm.
is very hard to justify, like because there is like I mean for like if it's say a very rare disease that only
Mm-hmm.
affected like very rare amount of people,
Yeah,
then there'd be a lot more discussion. But because there is that emergency that like people are
yeah,
drop that the there are like kind of that has to be taken into account. Um,
for sure.
so
But I mean, it
yeah,
is a fascinating topic. I mean, almost like outside of the scope of us talking about like how the research is done in general, like it brings in a lot of philosophy, even right,
yeah,
into this discussion, and a lot of just like the values of the IRB, right? The values that we have as a society, kind of get tickled a little bit by studies like this.
yeah, this is the sort of thing I had to deal with a lot in research integrity when I was working as one of the things we had to do is check the ethics statements and check whether a study would need it because I think from a journal's perspective it's also about like liability and making sure that these
Mhm,
questions have been asked, making sure that
mhm.
researchers have considered these and they have like an explanation that they give because they will be called up on this. People are going to read this and they're going to go, okay, why was this important? And they have to have something ready. They have to have some accountability as well.
Sure,
And I think
sure.
so.
Yeah, so I guess one of the interesting things if we chose this paper would be what is that ethics statement, right? And like reading it from that perspective.
Yeah.
Okay, let's
Okay.
move on.
Yeah. Okay. Analysis of SARS-CoV-2 mutations in the United States suggests presence of four substrains of novel variants, and novel
Yeah,
variants. Ooh.
I do think they're cashing in here on the news of all the variants, because essentially they're using just the GSAID data as we have access to as
Yeah.
being available throughout this pandemic, an amazing resource.
Yeah.
And they try to find what variants are emerging here in the states. And I like that they rank them against the variants that are appearing in the world, so you kind
Mhm.
of see that distribution. Um, and I think, yeah, I mean, I don't know, it's almost kind of boring from the sense of like, like okay, this is an analysis. I haven't. It's analysis someone was able to do with data that exists. Of course, we know that there will be variants. I mean, variants have been popping up around the world. Natural variation is something that happens during multiple infections, right? We've said it
Right.
many times, I think, on this stream, right? That like mutations happen, and when you have enough of them happen, there will be ones that are more successful than others. And so the primary way of making sure it doesn't happen is by limiting the spread. So anyways, back
Yeah.
on that
Definitely.
bandwagon.
Yeah, so yeah, this is a very sequence gazy paper where they download a sequence. So theoretically, anybody could have done this research, but they were the ones who decided to sit down and because you don't need much in terms of resources. You just need the software and the know-how. Um,
Mm-hmm.
so
Mm-hmm.
in this case, they downloaded these sequences from the database. They kind of put them together and they did some prediction modeling to determine the effects of those mutations. So they do a lot with this, with what they have, but
Yeah, absolutely. I like that they map them onto the structure of the proteins later in the paper. They have some figures where they map it on, and I'm not sure what these network graphs are, but they intrigue me. Like if there is information in there about maybe where I mean, my guess is that maybe I can try to read it. No, I don't understand what it means. The difference of subgraph centrality between the network with wild type and the network with mutant type. That's not off the top of my head information.
That's graph theory. That I don't think we were trained to in microbiology school, which I think we I can think more and more nowadays we are going to have to learn more about like computer algorithms to do our jobs because it's it's out there now, and we have to.
Sure.
It's not just about like do your plankton work, but actually understanding what other people are doing so that we can
Yeah.
figure out how to
Yeah.
apply it to
Yeah.
the kind of things that
A
we do.
huge thing. But yeah, so they must leverage some other data, right, to gain some insight into how this mutation stacks up with other mutations. Yeah, I mean I think that's all there is to say about this.
Yeah.
What's interesting is that they never really tell you, and this is going to be difficult, I think, in parsing all the research out there, and probably something that everyone is grappling with right now, is that there are these variants that have multiple mutations in them. There are papers that speak to subsets of those multiple mutations. um, and so there's it is not always evident when we're speaking about one of these papers, like what variant are we talking about, right?
Mm.
What locations are we thinking about? And like this makes it even more complicated because now we're saying across the United States there are locations with different variants, right? And yeah, I think it just shows us that there is a lot of stuff going on right now, and I don't know if people are doom scrolling the news a lot and seeing all this stuff about variants, but I mean, there are a lot of different types, and understanding them is not as easy as just like the named ones that have been going around. Those are the ones that I think have a lot of public health attention
Mm.
on them because they're becoming prevalent because there's initial data. But at the end of the day, like anything could emerge, right? Like there's the possibility of emergence. So keeping rates low is still very important,
Yeah.
even with the vaccine coming
Yeah.
out. Maybe even more so with the vaccine coming out, because as we talked about in the earlier papers, right, that could also be a selective pressure in some ways.
Yeah, exactly. And the thing also is like you need to keep in mind that a lot of these variants aren't going to be like lethal or have vaccine immunity. A lot of them are just going to be random. Um,
Yeah,
so
absolutely.
they,
They don't even right. They only show them and they guess what effects they will have. They can't say in this paper what these variants are going to do, right? And they can only guess about it.
yeah,
Educate, yes, yeah.
yeah. But I mean, it's interesting. This is one of those papers you'll read so you can be a hipster in about 3 months' time when you go, oh, I recognize that mutation.
Totally, totally.
I read it here first, so yeah, that's one reason to look at this paper so that we can be smug and like, oh yeah, we covered this mutant when it was, but back when it was just not being very popular.
Yeah, I mean, I already feel the smugness because we were talking about the D614G and the preprints that were coming out, right? That showed its biological significance, right? And that is one of the main mutations that is in a lot of the variants.
Oh yeah, I mean within the mink outbreak, we were talking about the N501Y, which is now like everyone's talking about it. But
Yeah.
yeah, I mean it's the bad kind of hipster thing where you like everyone's miserable and we're smug and being like, oh great, now this is like nobody's impressed with us at all. No one's gonna give us any credit for this.
I'm not even impressed with that.
Yeah, I'm not impressed with us. We're ghouls. This is terrible. Um,
Okay,
yeah.
this one we got
As-
is assessing the influence of climate on wintertime SARS-CoV-2 outbreaks.
Yeah, so this is interesting because it takes data from coronavirus outbreaks in the US and compares it to the weather data. Uh,
Mm-hmm.
so, so it's not just enough to compare like infections in areas that are different, as there can be so many confounders. So they look at humidity, they look at a bunch of different factors, and then they kind of put in a susceptibility, an SIR model, and use
Yep,
that to
the
estimate.
same model that people have been using. That's a very common, seems like epidemiological model we've been encountering.
I mean, it's the most simplest and it is the most broadly applicable to most infectious diseases. I mean, I mean, yeah, and I mean, so it's how you work out the R0 number, which is a basic reproductive number, and they've been looking at how that gets affected by different weather conditions.
Mm-hmm.
And it's interesting because they say that the weather might have an effect. Not huge because like the interventions are being put in. Lockdowns, kind of social distancing, tend to have a bigger role than weather. So
Mm-hmm.
they
I
basically,
like it. I think it takes down,
yeah,
like, to me, it's like, you can, from the scientific point of view, you're like, okay, this is fairly expected.
mm,
But I mean, compared to media coverage, right? Maybe someone is reading things and it's like, oh, don't worry. Like, I've heard that on the street. People are like, oh, like, it's gonna go down during this season because the weather. It's
mm,
like, well, okay, the weather, but the weather also impacts our behavior, and perhaps that behavior is a very important thing. Behavior is also modified in many other ways, right? Other non-pharmaceutical interventions. So yeah,
yeah.
weather's not that important.
I feel like since the discovery of central heating and air conditioning, the amount of the way the weather directly affects our health has changed a lot. It's more about behavior, and I feel like
Sure.
that's uh, an interesting thing to think about. Um, Speaking of behavioral interventions, we're looking at the impact of non-pharmaceutical interventions on influenza and other respiratory and viral infections in New Zealand. So this is yet another thing for New Zealand to be smug about. Um, yeah, so this isn't about COVID-19, but it's about the kind of outcomes of those lockdowns and the kind of border controls.
Hmm.
Yeah, this looks into influenza outbreaks and respiratory diseases. So it basically looks at how respiratory like illness compares like after the main lockdown went out, comparing it with last year. So I've pulled up some graphs. Like the red line is like this year, and the blue lines are like the previous years, and you can and basically like influenza has basically flatlined in New Zealand. There's not very many
Mm.
outbreaks happening. Uh,
I mean, they were very effective in their COVID-19 lockdown, right? Like, that was one of the success stories we were hearing globally, right? Is their ability to control that outbreak.
yeah, they went into lockdown and then they maintained some of the controls and they got really big, really tough surveillance going on as well. And
Hmm.
so, uh, yeah, a lot of these seem to be. So if I look at, yeah, a lot of these are set after lockdown. So you had like one, like relatively compared to say the UK, a very short lockdown that was very severe, and then after
Hmm.
that it persisted. So I wouldn't be surprised if influenza went down like before or during the lockdown. I wouldn't be surprised,
Sure.
but like that is maintained after. That's the bizarre thing. I wouldn't have expected. I would expect it to creep right back up afterwards because people are behaving
But
normally.
presumably people are wearing masks, right? Like, it's
Yeah,
all right, there's all these other interventions that happen after lockdown that are in place because influenza is a respiratory virus, and they're the same interventions, right? Apply to influenza as they do to SARS-CoV-2.
yeah. Exactly. So that's very interesting. It might be interesting, like, to think about how our behavior will change going forward and how that will impact influenza outbreaks. Uh,
Yeah, I mean, I think I may have said this to you like off stream or maybe even on stream, but like, I don't know if I'll ever go back to, like, I think masks will forever be a part of like, my life inside the city. I mean, not that I'm gonna wear one every day when I go out, but if I am feeling under the weather, if it is flu season, right, it might be the time to wear a mask, and it seems perfectly fine to me if it can impact public health outcomes.
I mean, I'll let you in on something. I was wearing a mask before the outbreak because I was paranoid about like facial recognition. So I'm
Sure.
pretty certain it's still gonna keep that up. But now I have another excuse, so people don't think
Yeah.
I'm a paranoid kook. But but yeah. Moving on to the next paper, the antigenic anatomy of SARS-CoV-2 receptor binding domain.
Mm,
So this study looks at the areas of the spike protein that antibodies can bind to. Uses X-ray crystallography and like cryo-EM to like map out the like kind of areas that are bound by the protein. Uh, and they too also, instead of using Squirrel, they use a male torso to look
mm.
at the spike protein. Um,
So wait, no, this is not. This is they're looking at antibodies binding, right, to
yeah,
the yeah,
yeah,
not to the receptor.
yeah, they're looking at antibodies binding, but is like to the set. I mean, the other one was also looking at, but this is yeah, this is mostly looking at uh what areas are susceptible, rather than looking at yeah, and so
This is
they
interesting because I feel like we've seen a lot. We've seen a lot of stuff that looks at antibodies binding, like all of the stuff about the ACE2 decoys, right? Like there was a lot of stuff there that was predicated on where the antibodies would bind, right? They did all those studies in yeasts and like showing where all the different regions. So is it because they're using convalescent serum in this one that makes it different?
Yeah, I think it's mostly the modeling, the X-ray crystallography, and the cryo-EM that they're using. That so they use convalescent serum. They pull out like a bunch of antibodies from that, and they use competition assays to figure out which ones bind to which areas are best, and then they
Mhm.
use that to do some modeling. So it's mostly about like the thing that fascinated me about this paper was the way that they use the mapping onto the structure of the protein. Um,
Yeah,
and
absolutely.
I'll be honest, like there's also the fact that they decided to represent it with like some man's pecs, like that.
Yeah, the torso. It also has a very nice like hand-drawn quality to this one,
Yeah.
right? Some nice pencil shading in and
Oh, they in their actual figure they've got a fully like 3D representation of a man's torso. That's
yes, I see that. And um, darn, these figures don't have the legends right by them, but
but
I'm
yeah,
assuming 2A.
this is 2A. Yeah, and they instead of the because you can still sort of see the squirrel shape in there, but they instead of like so
Yeah. I mean, it's just different, right? Like
yeah
the head is now the right shoulder or something. Oh, no, no, the head is the left shoulder. The right shoulder is the tail.
yeah
The right flank is back.
yeah
The neck is still sort of the neck of a squirrel.
yeah. This is you never know what you're gonna get in some papers. But
But again, the tool is because like to discuss like to discuss these regions, right? Like
mm,
where things are binding, it's easier to say these terms because we know the relationship in space between the chest and the right flank, but you wouldn't know the relationship in space, right, between the middle of the protein and the side that it has these particular residues on.
yeah, that's right actually.
Yes,
So
that's the reason, right? Like
yeah
it is crazy that they're using all these analogies, but it serves a purpose because it's confusing for scientists too to just speak abstractly about proteins rotating in space. But when you can tie them to something where we have intuitive understanding of the relative regions, then it just lets you talk about it easier.
yeah. So the next paper we're going to talk about is spike D614 gene mutation increases SARS-CoV-2 infection of multiple human cell types.
Hmm. This is cool. I mean, it's cell culture cell types that
Mm,
they're using,
yeah,
but that comparative, right? Comparing the different cell types and how they interact with this mutation, which is something that, again, very mechanistic, right? Kind of bringing us back down to what exactly are the mechanistic effects of having this mutation. And I think the fact that they use different cells is really cool, and I'm excited about this paper
mm,
because it gives you that another point of comparison, right? That like in certain tissues, maybe the mutation has a different effect. At least that's where they're sort of bringing their hypothesis down to.
yeah. That's it's interesting because I mean they do some look at the mortality. They look at how like it enhances or change how what changes how it affects receptor binding.
Hmm.
Yeah, and but also the sensitivity of spike protein to proteolytic cleavage. So they do cover a lot of stuff here. Um, yeah.
Yeah, I think the other reason I picked it out was because I had recently seen a paper from this group that did like a CRISPR scan,
Hmm.
and I recognized the first author. So I'm like, oh, this is kind of interesting. This might be something that we don't choose this week, but we choose later on if we're able to get somebody to talk to us about it. I think that would be kind of cool.
Yeah, that'd be cool. That'd be
Hmm.
kind of cool. I think the last one we're going to talk about is structural insight reveals SARS-CoV-2 ORF7A as an immunomodulating factor for human CD14 plus monocytes. So
Yeah, I don't remember talking about ORF7 before. I
no, me
think this is a
neither.
new ORF that in the mix of all the different proteins that SARS-CoV-2 makes. And I guess no surprise. Again, no surprise because, hypotheses don't come from nowhere.
Mm.
And knowing that the virus will do something to the immune system means that people are looking for, right, effects on the immune system from its accessory proteins. Um,
Yeah,
but yes. yet another immune modulatory activity of an accessory protein.
Yeah, I mean, this is like it's got like an immunoglobulin-like area in it, and they test it, like it compared with like the OR7A from original recipe SARS-CoV-2. Find this one is actually more immunomodulatory. So maybe half the paper is looking at the structure of OR7. So they do solve the structure, and they mix it in with like mononuclear cells to see which ones pop. They found that CD14 reacted to it. And so what's the reason for that? And they further characterize that interaction. So the interesting to me in terms of like OR7 isn't necessarily expressed. So the question is how to have those neutrophils, not neutrophils, monocytes in culture.
Right, because it's happening intracellularly, presumably, right? Unless it's somehow secreted by the cells. That is.
I mean, yeah, or I guess it would like if like there's necrosis or something like that where the cell just pops or releases
upon
stuff.
lysis of the cells, this gets released into the immediate area.
And in that case, which would be the first cells on the site? Well, it'd be monocytes would be the first cells on the scene to clean up a necrosis. So something that messes with that. So that's an interesting like line of thought for this.
Yeah, sure. Probably again, like it sounds like the way that they move from structure to their immune function, right? They then added in
Yeah.
afterwards. So there's something in the structure that made them interested, but then following up, it's like, oh, what? How would this affect, right? Like what we know about the pathogenesis.
I mean, yeah, it might not be the primary function during infection because sometimes proteins can have more than one function, and it could have a function internally in the cell as well. But
Absolutely.
but
And I
it's
mean,
an
knowing viruses sometimes multiple functions
yeah.
of protein is not an uncommon event.
Yeah, viruses they have very little. They have a very short like length of genome, so they need to do as much as they can with it, which is why they're kind of so fascinating to look at because they have lots of very interesting ways to organize a genome to get stuff like that done.
Totally, totally. Okay, so that's what we have on the docket for today. I guess
Yeah,
you got to choose something that we are interested in talking about. And again, we have the back catalog. I went
yeah.
I went through the back catalog and I removed the ones about organoid cultures because I think we have a much better organoid paper now that will stick with us if and when we decide to talk about that.
I
Uh,
mean, I'm quite happy with the organoid paper. I mean, I'd be happy to talk about that one next week. Because we haven't really talked about organoids that much, and because for me it was either that or the like modeling paper, but we did do modeling paper quite recently, so
Yes, we did do the modeling paper quite recently. Yeah, it's once again like choosing the modeling paper is one of those things where I feel like the world's attention is on like the outcome of these models, right? Like it's very much
yeah,
like, well, what do we make of all these things? So there's that. We already did
yeah,
once. I kind of don't want to necessarily cover something like that again. It's also
mm,
a lot to understand, maybe, which is not
yeah,
a good excuse. I'm actually interested in the mechanism that the RNA, the mRNA one, uh,
yeah, the
RNA
hamster one,
your COVID-19 model. Yeah. Right.
yeah, because that goes into
Like,
a
how does this vaccine work? I kind of want to know. And it's cool because they're using that. Like previously, when we saw single cell techniques, I think we saw them in the supplement. It was just like something
yeah,
added on in the background to say, like, oh, look, I think it was with the neuropilin papers where we saw it, right?
yes,
And there's
I think
like,
it
oh,
was,
we looked at big single cell databases and we confined neuropilin on these types of cells. And so here they actually did it for their experiment. So using the technique in service of experimental design, I think is something interesting to put out here. And again,
yeah,
where previously you would just take a lot of histology and maybe never get the full picture too after all of that histology.
right. Yeah, because yeah, you only ever get a small sample of histology, and it's only the stuff that actually survives the process because there is some like cut off cuts and other things, and some of cells get like destroyed.
We should
So
put ICS too much on a pedestal because, of course, we're still processing those tissues in order to get them to
true,
the thing that can go through the fax machine.
yeah,
Something.
but yeah, I mean you get like a sample of all the RNA like in the cells, and I feel like there is a lot that you can drill down to in from that.
Hmm.
Um, so yeah, I'd be interested in that one.
I feel like there's a lot of good ones in this in this mix. I am slightly. I'm not sure if you're excited about the PeriDomestic mammals one from the
I am
FAQ.
the opposite of excited for that. I
Yeah,
mean,
science I think is quite dry. It's more just oh, they actually captured all these animals. Why?
yeah, I feel I'd like pull out my animal ethics book and just like be reciting like all the well, all the things that I feel like all the things that I as like a journal publisher would be looking for in terms of the ethics to make sure that the journal be have its arse covered rather than like looking at the ethical issues because because when you're at that point you can't really go back to research go look you can't do this research can't be I mean there have been situations we have gone like no we cannot publish this it's just unethical but I mean it's very hard to go back because there is always this argument like, oh, we've done the research now, so may as well publish it. But then that publication
Yeah,
could
I see what
encourage
you're
other people
saying.
to do it.
Like by the time it gets to the journal, right? Like what's the point anyways, right? Because it had to be stopped at an earlier point if it was
Yeah.
impact, yeah.
So I mean, there have been times where I think we have like sent strong, like rejected and sent strongly worded letters like, don't do this, please, ever. But again, journals don't have much leverage on the universities
Yeah.
because they can always go somewhere else. They can. Here this is a preprint that there's and preprint servers don't have that they can't have that level of scrutiny that a journal has because they just have so many things. So that I mean
Okay.
yeah, I guess like if you want to hear me moan for about 2 hours about ethics, then
I,
this is a paper.
yeah, no, it was good. It's good to hear your perspective on. I did want to elicit more thoughts about it. But yeah, I don't think
Yeah,
we don't have to do it.
I mean yeah, I
The other things in the past we had, I guess there's still the real-time conformational dynamics of SARS-CoV-2 on virus particles, where they use FRET to see how the spikes are moving
mm,
on viral particles. That's another one that we can throw in.
yeah.
And I think to me, that's a good mix of things. What are you feeling after this?
So
Running through some.
after this, I'm bouncing. So I'm feeling the hamster one a lot. I'm feeling the kind of detailed analysis of what the cells are doing. I
Gotcha.
feel that's like because again, it's got the things that people are quite interested in, like vaccinations, some details about how vaccinations work. I
Mhm.
think that's.
Absolutely.
I
Some people
think
have this. Some people have this mRNA in their bodies right now,
yeah,
and
exactly.
of course, don't pulverize human lungs to find out which cells are being affected by it. Yeah.
Yeah, exactly. So we don't have to think too. I mean, yeah, and we can talk about ethics issues because it's hamsters. I mean, we can still have a conversation about that if
The
necessary.
thing that I realized would be interesting about it, I think we should do it, and I'll leave you with a thought that it's also interesting because this is a new vaccine strategy.
Yeah,
And so I actually don't really. I wonder whether or not there'll be any information about how this type of vaccine works, right? Like, will they know what cells took up this mRNA and ended up
Th-
expressing the spikes? I'm not sure.
I mean they better because if they're sequencing all the RNA in these cells, then they surely should be able to see where the SARS-CoV-2 is,
Yes,
and also compare that with the vaccine because Moderna vaccine like RNA isn't actually exactly the same as the SARS-CoV-2. There are some specific changes they've made.
right.
So if they
They
had a way
yeah, yeah,
So, because there are some like alterations they made to make it more stable. So the question is whether they've got sensitive enough techniques to spot that
yeah,
or
yeah.
not. Um,
Yeah, I'm
so
down for that then. I mean, it's a preprint, but this is this peer review process, right? Like we're doing peer review here. Maybe not
yeah,
the same experts that would get it, but we have our own perspective, and I'm curious. I want to know.
yeah. I mean, we do try to go into as much detail as we can, and obviously we are enthusiasts. We're not necessarily experts. So, and
Right.
yeah, we've talked about preprints before, and I think this if you have that asterisk in your head, like saying, okay, I think you should, but firstly, I think you should have all papers of like maybe like think about what this is actually telling us.
Yes, absolutely. Yeah, you always have to have your spectacles on.
Yep.
Skepticals, skepticals.
Skeptical.
Okay, so join us next week for our deep dive where we're going to cover mRNA 1273 efficacy in a severe COVID-19 model, attenuated activation of pulmonary immune cells after challenge.
And we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it is possible we didn't get everything right. So science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
Yep, I totally agree. As I said earlier, this is the peer review process. We hope that everyone can participate. And so reach out to us over Twitter or the hashtag #MicroTWJC to ask your questions as you read along.
Yeah, yeah. Please let us know. It's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content. We'll see you then.